Hello and good evening students. How are you all? I hope you all are doing fine, and I am your biology mentor Aafeeyat Khan, and I am here with one more episode of the chapter The Fundamental Unit of Life from class 9 series, where we will be completing our term 1, that too like in no time. Okay, so just give me a thumbs up in the chat section that I'm clearly audible and visible, so we are good to go for the classes. Okay, let me also quickly check. Okay, so I guess we are clearly visible and audible, so we are good to go and we can move forward with the classes. Okay, so moving forward with the chapter, the last chapter where Priyanka ma'am, the OG chemistry mentor, she explained how the evolution has contributed, not just the evolution, how the structure of atom has contributed to the whole life mechanism. Like when the atoms came together to form element, then element to compound, and then compound came together to form a living material, a living structure that was the smallest, tiny little structure, that was cell. Yes, I am talking about the origin of life. Until here that was chemistry, but we had to learn about how this structure has evoluted, or like how the process of evolution and how the chemical things and everything has contributed in the formation of living organism, that is us and plants, animals, and all the other living organisms. So we have studied that a single cell is responsible for the formation of whole organism, like any kind of organism. Okay, a group of cell will be forming tissue, and a group of tissue will be forming organs, and the organs will, a group of organs, organ system, then the whole organ systems, like different organ systems will be forming one organism. I am talking about all the organisms, plants, humans, and animals. This we have studied in the last chapter. Then we came up this we have studied in the last class of this fundamental unit of life part 1, where I have told you that the discovery of cell was done by Robert Hooke, but he didn't discover the living cell. So he discovered the dead cell, and that too in the cork of come on, and in the cork cells, where he had studied this, the cork cell or the cork of a plant or any bark of a tree, that consists of a group of cells, and that cells he declared that these are the dead cells because they were not moving. The movement was the first criteria to consider anything that is living or non-living. So he considered that the cells are not moving, then they are dead. Okay, then comes the Leeuwenhoek, then comes another scientist, that is Leeuwenhoek, and he discovered that the cells are not dead, they are living. And how did he concluded that the cells are not dead, they are living? Similarly, on the basis of movement, how Robert Hooke discovered that the cells are not living, they are dead. Similarly, if I talk about the vice versa, he did. If cells were moving, then they are considered as dead, and he named them as animalcule. And that's why for the discovery of cell, he is also known as the father of microbiology, because he discovered the very teeny tiny little thing, that is cell. Okay, moving forward, there comes different scientists who came up with different theories and everything, but there comes, how do I know how the cells came from? Like there comes another theory from two scientists, that is Clayton and Swan. That was the two biologists who gave the theory. Any organism, any kind of organism is formed of cell and the cell products. This was the cell theory given by Clayton and Swan, and later on it was modified by Rudolf Virchow in the year 1855. He said omnis cellula is cellula, because till now I don't know from where cells are coming from. Now I know cells are coming from pre-existing cell, like any pre-existing cell is forming new cells. So all new cells arise from pre-existing cell. This was the modification given by Rudolf Virchow, and the cell theory become modern cell theory. Okay, then moving forward, we had discussed about the different shapes of cell, like depending upon the functions, the cells have different shapes, either regular shape, fixed boundaries, or irregular shape, no fixed boundaries at all. Then we have discussed about the sizes of the cell. Sizes of the cell, talking about the smallest tiny cell, that is mycoplasma bacterial cell, which is about 0.1 to 0.3 microns, and talking about the largest cell, that is ostrich cell. Largest means I am talking about the diameter, okay, the length and the breadth, both I am including, okay. Largest cell is the ostrich egg. Then talking about the longest cell, that is the neuron cell, because we have to transmit the signal throughout the body. So a longest cell was neuron cell or the nerve cell. Then we have studied about the different cells, although that RBC cell, then we had sperm cell, ovum cell and multiple cells. So till now we have studied that we have multiple types of cell. Now I want to know what are the types of cells. Okay, now I want to know what are the types of cell. So for the types of cell, we know that there are now multiple types of cells, but depending upon what basis. So we had two bases to classify how the cells are made and what is the type of cell. So we have two bases, on the basis of nucleus and on the basis of cell wall. If I describe on the basis of nucleus, we have two types of cell, that is prokaryotic cell. One is prokaryotic cell, okay, prokaryotic cell, and the other is eukaryotic cell. Eukaryotic cell. Okay, so we have two types of cell on the basis of nucleus. If the nucleus is primitive or not defined at all, that is a prokaryote cell. Okay, let me describe you this way. Okay, prokaryote. First, let's talk about the prokaryotic cell. Talking about the prokaryotic cell, here the pro means primitive. Here the pro means primitive, and karyotic means the karyotic word is coming from the word karyon, and karyon means nucleus. So if I have to talk about any primitive nucleus, I am talking about prokaryotic cell. But with respect to the prokaryotic cell, if I have to talk about any new cell, then or any new nucleus or the true nucleus. So here U means true. Here U means true, and again karyotic means nucleus. So if any of the cell has a true nucleus, that means I am talking about a eukaryotic cell. So what is the difference? The basic difference between prokaryotes and the eukaryotic cells are the nucleus thing. Okay. So first let's talk about the nucleus only. So if a nucleus is not well defined, means it does not have the boundaries, it does not have proper chromosome or the genetic material, then what it is, it does not have any proper boundaries or anything, then it is not a nucleus. Then what do we call it? We call it as nucleoid. So they do not have proper or well defined nucleus. No well defined nucleus is present. So here well defined nucleus is absent. So what is present in respect to this? A nucleoid is present. What is present, student? A nucleoid. So the name of the structure which is present instead of nucleus, that is nucleoid. But if I talk about eukaryotic cell, they have well defined nucleus with double membranous structure. They have double nuclear membrane. So here there are two nuclear membranes which are present. Okay, now moving forward with the genetic material. So they have single chromosome, and these have more than one chromosome, because this is a primitive nucleus, so they don't have more amount of genetic material. So these have single chromosome. But if I have to talk about eukaryotic cell, they have more than one chromosomes. Okay, moving forward with their sizes, if I have to talk about the size of prokaryotic cells, they're very tiny organisms, or they are tiny cells. So their size ranges from 1 to 10 μm, that is micron. And if I talk about the size of eukaryotic cell, then their size is from 5 to 100 μm. They are very large as compared to prokaryotic cells. Then talking about other membrane bound organelles, obviously if the prokaryotic cell is present, then it don't have membrane bound organelles. There won't be any proper well defined boundaries or anything. There won't be any division of labors as compared to the eukaryotic cell. But if I talk about eukaryotic cell, yes, eukaryotic cell will be having membrane bound organelles. It will be having division of labors because it is more modified and new and true form of cell. So if I talk about prokaryotic cell, there are no membrane bound organelles. That membrane bound organelles are absent. And if I talk about here membrane bound organelles are present. Okay, so this is the thing for prokaryotes and eukaryotes. Talking about beta prokaryotes, then all the bacteria and everything are included. All the bacteria are included in prokaryotes, and eukaryotes, other than bacteria, everything is eukaryotic cell. Okay, moving forward to the other type or the other type of classification, what we have to deal, that is on the basis of cell wall. If any cell has cell wall, that is different, and if any cell does not have cell wall, it is different. So mainly we are classifying on the basis of cell wall. Then we have two types of cells, that is plant cell and animal cell. Here plant cell, they do have cell wall, and if I talk about animal cell, they do not have cell wall. The outermost membrane of plant cell is cell wall, which is formed of cellulose. Cell wall is formed of cellulose. But if I talk about animal cell, their outermost membrane, is cell membrane. Their outermost membrane is cell membrane or the biomembrane, which is formed of lipids and proteins, that we will discuss later. And if I talk about other differences also, these plant cells, they do have differentiated structure to form food, because they are producers. I told you about this, that plants are the producers and animals are the consumers. So plant cells do have a specialized mechanism for the preparation of food, that is presence of plastids. So plant cells have plastids. Here plastids are present, but if I talk about animal cell, here plastids are absent. Okay, here plastids are absent. Okay, so now we know that there are two types of cells. Like if I have to divide the cell on the basis of something, I can divide the cells on the basis of two things. One is nucleus and the other is cell wall. On the basis of nucleus, I have two kinds of cell, that is prokaryotic cell, that is primitive cell, and the other is eukaryotic cell, that is true cell with the true nucleus. Okay, if I talk about on the division on the basis of cell wall, then I have two types of cell, that is plant cell and animal cell. Plant cell have cell wall and they are the producer, so they have differentiated structure known as plastids, where chloroplast will be present, which consists of green color pigment, that is chlorophyll, which is responsible for the trapping of light. Then the whole process of the whole mechanism of photosynthesis going inside the chloroplast. Okay, then talking about animal cell, they do not have cell wall, they do not have any kind of specialized kitchen, because they are not the producer. If they are not the producer, why do they need kitchen? Obviously, if I am getting food to eat like anything, I will simply eat. Why do I have to build or waste my money on building the kitchen also? So it's like very simple. If they are not the producer, they do not have the kitchen. Simple as that. Okay, moving forward with this, that what is a cell made up of? Now I want to know, if I'm talking about different kinds of cells, different like on what basis I'm dividing and everything, if I want to know that, now I want to know what is a cell made up of. Like there will be something which a cell is made of. So now what a cell or the structural organization, how I will differentiate a cell structure in different forms. So if I have to differentiate, or if I have to say what the cell is made up of, I can say a cell is made up of four things, basically four things. Okay, one is cell wall, like the outermost covering. One is the cell wall, which is the outermost covering. If I talk about only in plant cell, okay, that is found in only in plant cell, as you can see here, only found in plant cell. So outermost is cell wall. Then little more, okay, plasma membrane, which is found everywhere. So we have the second layer, that is plasma membrane, or I can say it's cell membrane or biomembrane, that I will deal later. Okay, then apart from that, we have a covering. Now I have to fill a fluid inside that, and that fluid is known as cytoplasm. So it is filled with the fluid, and that fluid is known as cytoplasm. And now a cell has already the cytoplasm filled in, the fluid filled in. Now I have to insert the other organelles, or I have to make the division of labors. Like I want to know what part is performing what function, because everything cannot perform same function. And if I talk about, okay, let's talk about if you want to run any organization, okay, if you want to work in an organization, or if you want to run an organization, you have to divide separate branches, or you have to divide ki authorities ki, this person will be doing this part, this person will be be doing this part, this person will be doing this part. So you have to differentiate, or if you have to distribute the roles and responsibilities among each and every one, so that there won't be any clash or there won't be any mix of communication or anything. So when you distribute the roles and responsibilities among different people, that will be more productive, and the time consumed will be less. So that's what the division of labors. And for that division of labors, here the employees are known as cell organelles. So here we can see different cell organs like nucleus, mitochondria, and endoplasmic reticulum, Golgi apparatus, then we have centrioles, then we have other like ribosomes and other cell organelles. So these are for the division of labors. I can say ki I have mazdoor work, so I have labor thing. So that labor and those people who are working for this whole organization, that is cell organelles. Now let's study one by one for how, like what cell is made up of and everything. I want to know in deep that what cell is made up of, what is that function, what is the characteristic feature, everything if I want to know. Let's quickly jump to the brief introduction about each and everything. What is a cell made up of? So the first thing which is omnipresent in all the cells, I'm talking about all the cells, okay, that is plasma membrane. So basically plasma membrane is the outermost covering for all the cell, that is plasma membrane. A plasma membrane is also known as cell membrane, and it is also known as biomembrane. Okay. Now, why this name has been given? Let's differentiate one name, and we will get a clear picture of this plasma membrane, what it is. If I talk about plasma membrane, plasma is any kind of fluid. Plasma means any kind of fluid. So any kind of fluid which has been incorporated or which has been covered by a membrane, that means I'm talking about plasma membrane. So plasma here means any kind of fluid. So what fluid? Like a cell is basically a closed vessel, and I have put all the fluid at the vessel. The boundaries which I have made from the vessel, that is membrane. So what will I say for that membrane? That will be a plasma membrane, because a membrane which is covering the whole plasma is known as plasma membrane. Okay, the next term is cell membrane. I am talking about a structure called cell. So any membrane outside the cell is known as cell membrane. Now coming to the third name, that is biomembrane. Bio, we all know, bio means life. Bio means life or living. So any membrane which is living in structure, that is known as biomembrane. And why am I talking about this? This plasma membrane is a living membrane, because this plasma membrane is moving. It is constantly moving. It is changing its shape, structure every time. So that's why I am saying it is living membrane, because we have studied anything which is moving is living, and anything which is not showing movement, that is dead. So if I talk about plasma membrane, so plasma membrane is the outermost covering. Okay, what is plasma membrane? Plasma membrane is the outermost covering of almost all cell. And what it is made up of? If I talk about the composition, if I talk about the diagram, first let's talk about the composition. Okay, if I talk about the composition, it is formed of lipid and protein. It is formed from lipid and protein. So lipid is basically present in bilayer. Like there are two layers formed of lipid, and protein is basically inserted. Protein is basically inserted in those two layers. Like when you look at the diagram, this is the layer. This is basically a layer of lipid. This is one layer. This is one layer of lipid, and this is second layer of lipid. This is basically second layer of lipid. So we have bilayer lipid. We have bilayer of lipid, like two layers of lipid, and proteins are basically inserted in it. So proteins are basically inserted, as you can see here. So this is protein. 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 So basically this protein are inserted in the lipid layer. So we have we have 2 layers of lipid and proteins are inserted in the lipids. Okay, so this is protein. Okay. Now, talking about the structure of plasma membrane and the lipid, we have 2 ends of lipid. So, if I closely observe the structure of lipid, it is the head and the tail. Okay, why we have head and tail? Because we have, any cell has 2 environment. Like if I talk about this cell, it has like the external environment, and the inner environment. So, the external environment is different and the inner environment is different. External environment also has the liquid flowing outside it, and internal environment also has the liquid flowing inside it. And if I talk about the middle of that the layer, then there won't be any water content. So what plasma membrane is doing, it is separating the external environment from the internal environment of the cell. So what plasma membrane is doing, plasma membrane is basically separating the external and the internal environment of the cell. Like I have to keep the environment separated from outside to the inside, and all the movement, like all the the transportation mechanism will be through this plasma membrane. So basically this plasma membrane is like a sheet. Like if I have to transfer one thing from this side to this side, I have to cross this barrier. And this barrier is a plasma membrane. Like one thing is transferring from this end to that end. So that barrier will be plasma membrane. This is external environment, that is internal environment. So any kind of particle which is traveling from external environment to internal environment, it has to pass through the plasma membrane, and anything which is transferring from inside to outside, that has to transfer by plasma membrane. So plasma membrane is basically a barrier between the external environment and the internal environment. All the transportation is going through this plasma membrane. So if I talk about the structure of lipid, they have basically the heads and the tails. Okay, so what are they formed of? Like lipid, any kind of lipid is having these kind of tails. Like this is a head, but this head is at the side where the water is present. If I talk about the external, here water is present. If I talk about the internal, here water is present. So head is present at the outside and head is present at the inside. Like if I talk about, these are the 2 sides. So this is a plasma membrane structure. So this is connected to the outside of the cell, and this layer is connected to the inside of the cell. So all the heads of the plasma membrane will be lined like this, like the heads, and here also heads, and the tails will be facing towards each other, so that they don't have any contact from the water. So that's why we have a name for the head, that is water loving head. And if I talk about the tail, they are water hating tail. They are water hating tails. So this is the structure of plasma membrane. Now, what kind of transportation mechanism this plasma membrane is forming, and the structure, okay, first let me talk to you about the structure. This structure was given by the scientists Singer and Nicholson. Okay, this is basically the structure is known as the fluid mosaic model, and this was given by Singer and Nicholson. This is not mentioned in your NCERT. This is just extra information. If you want to grasp it, it's completely your choice. The name of the scientist and the structure, that is completely your choice, students. Okay, that is not a burden or something that you have to remember their name. No, it's not. It's just your choice how you want to do it, how you don't want to do it. Okay, so if I talk about this structure, this structure is the fluid mosaic model, which was given by 2 scientists, that is Singer and Nicholson. Singer and Nicholson. Okay, that's completely up to you. Now let me tell you about the transportation mechanism through the plasma membrane. Okay, Now if I tell you how this transportation mechanism happens, or what happens when I put animal cell or any plant cell in the water, and that water is not just water, that water is either the sugar solution or the salt solution. So what will happen, like if I have two kinds of solution, okay, one solution is, this one is the salt solution or the sugar solution. Okay, this is salt or sugar. This is also salt and sugar. Okay, these are the solutions. What will happen if I put any plant cell or any animal cell? This is a plant cell. What will happen, or what changes I'll observe if I put the plant cell or the animal cell in the salt or the sugar solution? Is it easy that I am talking about only one kind of solution? Like okay, if I am making any if I want to make a lemonade, is it possible that I'll make a lemonade in a fixed quantity every time? No, it's not possible, because if I want to make a lemonade, I can be sometimes I can put more salt and sugar, or sometimes I can put less salt and sugar. The water quantity will be more. So we have two kinds of solution. Basically what we have is not two but exactly three we have. Okay, but let me talk to you about two solutions where the changes can be observed. So I have two kinds of solution where I have put extra salt and sugar. Okay, where my salt, let's talk about salt. Okay, where my salt is more than the water, and that solution where the salt, okay, let me tell you what is salt and solvent. Okay, first let me tell you what is a solution. So a solution is a mixture of two things, solute plus solvent. So basically solute is something which has to be mixed, and solvent is something in which I am mixing the solvent. So here if I talk about this, then salt or sugar will be the solute, okay, and solvent will be water, and water is also the universal solvent. Okay, so here solute will be salt and sugar, and the solvent will be water. So what will happen if my solute, that is salt is more and water is less? This type of solution will be known as hypertonic solution. This type of solution will be known as hypertonic solution. This is my first type of solution. Okay, now what will happen if I add less salt and sugar and more water? That type of where my salt is less but water is more, that type of solution is known as hypotonic solution. This is my second type of solution. Third is there, that is isotonic, where salt and water will be equal in quantity, and that type of solution will be known as isotonic solution. Okay. Now what will happen if I put animal cell or plant cell in any of these three solutions? What will be the changes I will observe? Now quickly look to the observation what I will observe. Okay, so if I talk about a hypertonic solution, where hypertonic solution means what? Where my solute is more and solvent is less. Okay, let me just quickly change the color so that it will be clearly visible. Okay. Hypertonic solution is where my solute is more and solvent is less. Isotonic solution, where my solute is equals to solvent. And talking about hypotonic solution, where my solute is less and solvent is more. Okay, now what will happen? So here when the concentration, when I'm talking about solute, I'll be talking about concentration, where I'll talk about solvent, I'll be talking about potential. Okay, understand? So where my concentration of the solution is more means my solute is more. So if I put animal cell for like longer time, and I can see okay, so if I put animal cell in the concentrated solution, that means a cell will be shrink like all the water, because what happens is the movement of concentration will move from higher concentration to the lower concentration. It happens in each and every case, and that phenomena is known as diffusion. That phenomena what I am talking about is diffusion, where where any concentration is moving from higher concentration. Okay, I have made the sign and I have written the sign. I have written about it. Okay. So basically diffusion is a process where the movement of particles will be from higher concentration to the lower concentration. So when I put in the hypertonic solution, the concentration of the solution, the concentration, like if this is the solution, if this is the solution, then the concentration here is more, and I guess I have made something wrong. Yes, so basically now it will be fine. Okay, so when I put any cell in the concentrated medium, like any cell in the concentrated medium, then the whole thing will be moving from higher concentration to the lower concentration. So hypertonic solution, everything will be coming out from the cell, like the water will be moving from higher concentration to the lower higher potential to the lower potential, and that phenomena will be osmosis. Same goes for diffusion also, but if I talk about osmosis, that means I am talking about the water movement. If I talk about the salt and sugar movement, that is diffusion. But if I talk about this, then I am talking about osmosis. Okay, let me just write down osmosis only, because you will get confused with this diffusion, because both the phenomena cannot be explained together. Okay, let me just put it down with the osmosis thing. So here what is happening is a movement of water from higher potential or higher concentration level to the lower concentration level. So higher concentration is inside the cell and the lower concentration is in the solution, because this solution is a hypertonic solution. Matlab solute is higher than the solvent. So if solute is higher and solvent is less in the solution, so here the concentration of water will be less and the concentration of water here in the cell will be higher. So the movement of water will be from inside to outside. So if I like this is my particle, and if I have to squeeze the water out, how it will be like I am squeezing the water out from the sponge, that will be shrink. So if the movement of water is from inside to outside, the cell will shrivel or the cell will shrink. Okay. Now if I keep the same cell, if I keep the same animal cell in the isotonic solution, then there won't be any changes, because the concentration here and the concentration here is almost equal. So if there is movement from outside to inside, then there will be movement from inside to outside also. That's why the shape of the cell will be normal and similar or the same structure. But if I keep the same animal cell in the hypotonic solution, where the solvent in the the concentration in the outside environment will be higher and the concentration in the inside will be less, then there will be movement from outside to inside to fulfill that thing. So the cell will swell, or we'll say it lysed cell. If I talk about the same situation with the plant cell, I won't see clearly visible differences from outside, because there will be cell wall protecting that cell. That won't be clearly visible. But if I talk about animal cells, they don't have cell wall. That means I'm talking about only the plasma membrane, and that will be clearly visible to me. But if I talk about plant cell, it won't be visible to me only because of the cell wall. Cell wall is rigid in nature, it won't show any changes. But that doesn't mean plant cell is not changing. Plant cell is changing. But where? Plant cell is changing in terms of this plasma membrane. You can see the changes in the plasma membrane. Like if I keep the plant cell in the same hypertonic solution, the plasma membrane, the gap between the cell wall, you can see the gap between, this is my cell wall, This is my cell wall and this is my cell membrane. So the gap between the cell wall and the plasma membrane has increased. Like the cell has been plasmolized. Cell has been plasmolized, like it has like this is my cell and these are the walls. So it has squeezed up and released from the wall. So the distance between the cell wall and the cell membrane has been increased. But if I talk about isotonic solution, there won't be any change and the cell will be same, equal. But if I talk about hypotonic solution, then how animal cell is swelling in size, this plant cell will also increase. It will generate a pressure, like all the water will be filling inside the plasma membrane. So it will create a kind of pressure on the cell wall that it has to be ki I want to get out from this. So that's why it will create kind of a pressure in the cell wall. Like you can see the distance, this and this. The cell wall and the cell membrane has been decreased to the next level and it has been creating a kind of pressure on the wall that I want to just get out from there. I want to burst out, and that condition is known as turgid condition. That in that condition cell will be known as turgid cell. Okay. So basically what's happening here is the process of osmosis. Now what is osmosis? Basically osmosis is the movement of water through any selectively permeable membrane. Selectively permeable membrane means, so if I talk about plasma membrane is a selectively permeable membrane. That's why it was showing the phenomena of osmosis and diffusion. So this is basically my cell membrane, which is selectively permeable. Selectively permeable. Selectively permeable means it is allowing only selected particles to move across the cell. Move across the cell means from outside to inside, from inside to outside. That's how we explain the phenomena of selectively permeable. Like if I want only few particles to enter in the cell, only it will allow the identified and the other requirements only. Like if I don't need something, why will I allow anyone to enter in my house? If I don't want anyone in my house, I'll simply kick them out. That's how the plasma membrane is working. What they want inside the cell, it will allow the plasma membrane will allow that to enter inside the cell, and if they don't want anything, there's something is extra, they'll simply kick off. That is selectively permeable concept. And the osmosis is completely working on that principle, that if anything that is transferring from selectively permeable, that too in a concentrated way, ki this much I require, I'll allow only this much to enter inside the cell. That is the mechanism. So osmosis is basically the movement of water. Osmosis is basically the movement of water. And the movement of water from a selectively permeable membrane. Like if I talk about this, then here the water, like here, okay, let me take not the water, let me just write it down as solvent. So if I talk about here, the pure water has more concentration of solvent, and if I talk about a solution, it will have less concentration of solvent. So we have studied that it will move from higher level to the lower level through a selectively permeable membrane. That's why from this higher level, it is moving to a lower level, that too through a selectively permeable membrane. And the movement of water molecule through a selectively permeable membrane from higher to lower level is known as, this phenomena is known as osmosis. This phenomena is known as osmosis. Okay, moving to the next, we have cell wall, because cell membrane we have completed. If I talk about cell wall is not a living structure. Cell wall is a dead structure. Cell wall is a dead structure because it is rigid. It is rigid in nature. It is rigid in nature. Rigid means if I try to change the shape of cell wall, then everything will be broken down. The cell wall has a rigid nature. And okay, we have a question, I guess. How osmosis can, okay, let me just check in my phone. Okay, I cannot see the question. Beta, if you are typing the question now, just let me see the question so that I can answer. How will I answer if I don't know the question? At least let me see the question, only then I will be able to answer. Okay. Hello ma'am, thank you so much. Okay, let's continue the lecture. So I was talking about the cell wall. That cell wall is a rigid and dead in structure. It is rigid in structure, that's why it is dead. Because if I try to change any shape or the structure, it will simply break apart. And we have studied from the last class itself that if anything which is not moving is considered as dead. If anything which is moving considered as living. This is the first criteria of the scientists where they have put that something is living or something is dead. This is the first criteria. Okay. Moving forward, talking about the rigidity, the cell wall is basically the outermost covering. Cell wall is basically the outermost protective covering, because this is mainly for the protection. This is mainly for the protection. Any cell which is having the cell wall, that means that extra protection is required. So cell wall, what it is doing is, it is there for extra protection mechanism. If I talk about plant cell, then plant cell are not moving, so they need extra protection. Like if I am saying plant cell is not moving, I have to go there and I can simply do anything to that plant. No, they have a mechanism, or they have a defense mechanism. The first line of defense mechanism, that is their cell wall. Animal cell that is living and that is moving, they do not require any extra mechanism because they are moving. They can move simply here and there on their own, so they don't need that extra mechanism. But whereas plant cell, bacteria cell and the other cells, they need the extra defense mechanism, that is extra covering on their body, that is cell wall. So basically cell wall is the outermost protective covering on them. And not all the cells, the important point is, not all the cells have cell wall. Not all the cells have cell wall. Only few cells have cell wall, like animal cell. Animal cell does not have cell wall. Plant cell, yes, they have. If I talk about plant cell, yes, plant cell do have. Plant cell do have. And what their cell wall is made up of? Cellulose. Cell wall is made up of cellulose. Okay, I have made cellulose. Then talking about fungi, they too have cell wall. And what their cell wall is made up of? Chitin. Okay, talking about bacteria, yes, they do have cell wall, and their cell wall is made up of peptidoglycan. Okay, I should not be saying the name, but this is just extra information for you to understand that the cell which require extra protection and extra defense mechanism, because all these things are not movable and everything in the environment, so they need some little extra protection, that's why they have cell wall. But if I talk about animal cell, no, animal cell does not have cell wall. Animal cell does not have cell wall. Okay, so cell wall is basically for the protection. It is a mechanical barrier, or it is a barrier between the external environment and the internal environment for the protection mechanism. Moving forward from the cell wall, we have nucleus. If I talk about nucleus, nucleus is what? The central, the mastermind, the brain of the cell. So nucleus is also known as the brain of the cell. Nucleus is also known as brain of the cell, because it is the mastermind which is controlling all the activities of the cell. So nucleus was discovered by, if I talk about the discovery, nucleus was discovered by Robert Brown. What did he name? He named it as carrion. He named it as carrion. He didn't name it as nucleus, he named it as carrion. That's why we are using prokaryotic, eukaryotic. That carrier word is from where? Carrier word was given by Robert Brown for the nucleus structure. Now what is this nucleus doing? Nucleus is basically a centralized. The first thing which was discovered after the cell wall and the plasma membrane was nucleus, because if I put the stains and everything like methylene blue, iodine, saffron and everything, like if I put all the stains now, the first thing that will be visible will be center of the cell. Like if I have to stain the cell, and this is my cell, and the first thing which will be visible will be the central line, and that central line which I can see here is what? Is nucleus. That is nucleus. So that's why the nucleus was the first thing, or the first structure to identified after the cell wall and the plasma membrane. Okay. So after identifying the nucleus, what we did was, what is a nucleus made up of, like what is nucleus doing there, what is the mechanism of nucleus, what all things are included in the nucleus. So nucleus is basically a double layered or the double membranous structure. Nucleus is basically a double membrane bound organelle. Nucleus is basically a double membrane bound organelle. What it is doing is, in that double membrane bound, as you can see, this is a nuclear membrane. This is a double membrane bound and the membranes are nuclear membrane. This is a nuclear membrane. But if I talk about the nuclear membrane, there is a pore, because if I want to travel from outside to inside and from outside to the inside the nucleus, I need a proper space to travel, because if you have double layer, the transportation will be difficult. So for the transportation, there is gap between, this double layer, like this is double layer. So there will be gap between this double layer, that will be, okay, that will be gone. Okay, I need to make it again. Like this. So these are double layered membrane bound. So we have a gap between these layers, and the gap will be nuclear pore, and this gap will be pore like structure for the transportation inside and outside the nucleus. So these pores are basically for the transportation. These pores are what they are doing is transportation from inside the nucleus to the outside nucleus. Okay, so what do we have in the structure? We have nuclear membrane, that is double wall layer. We have pore for the transportation. What do we have next? We have nuclear pore. We have nuclear pore. What they are doing is basically the transportation. They are doing the transportation mechanism from outside to the nucleus, from cell to nucleus, nucleus to cell. That is nuclear pore duty. Now what is the fluid? Like we have a fluid filled in the cell also. So we have a fluid filled in the nucleus also, and that nucleus is known as, the fluid is known as, the fluid filled in the nucleus is nucleoplasm. That is the fluid filled in the nucleus. So this whole fluid is known as nucleoplasm. This is nucleoplasm. Okay. Now moving further, if I talk about what is this nucleus, basically why am I calling this as a brain of cell? Like why am I saying the control center or the brain? Because it is controlling all the functions. The brain of the cell, because it is controlling all the cellular activities, all the other functions and everything. The brain of the cell, because it is consisting of genetic material, and that genetic material in the form of chromosomes. So basically this nucleoplasm will be consisting of chromosomes. So chromosomes are basically the rod-like structure. Which are formed of DNA and protein. So basically chromosomes are rod-like structures made up of DNA and protein, like this. This is basically a chromosome. It is made up of DNA and protein. DNA inside it, and it is packed with protein. So basically a chromosome is a rod-like structure packed with DNA and protein. It is a genetic material. DNA is basically a genetic material which is transferring from one generation to another generation. So the whole mechanism for the formation of DNA, the replication, the everything, anything for the formation of genetic material will be happening inside the nucleus and the cell. So where these things are, placed, that is inside the nucleus. So this nucleus is consisting of chromosomes. So these chromosomes are responsible for the generation to generation transfer of genetic material, the information basically. Okay. Now moving forward, this nucleus is a double membranous structure. Fluid filled is nucleoplasm. Then it has the genetic material. The genetic material is present in the form of rod-like structure, like packed in a rod-like structure, that is known as chromosomes. which will be transferred from one generation to another generation. If I talk about the discovery of nucleus that was discovered by Robert Brown, and he named it as carrion. He didn't name that nucleus, okay, he named it as carrion. Okay, now the information which is left, nucleus also consists of one more organelle packed inside it, and this is, okay, let me just change the color quickly. So this is another organelle which is present inside the nucleus, that is nucleolus. That we will study in later classes, but that you should be aware that nucleolus is another cell organelle which is present inside the nucleus. Okay, that we will study in later classes. So today we are running little late and we're a little out of time. So the rest of the things apart from the nucleus and the other cell organelles and everything we will study in the later upcoming classes. Okay. Now talking about the functions of the nucleus, the nucleus is responsible for the cellular reproduction. Cellular reproduction I have already studied cellular reproduction. Why cellular reproduction is happening? Because of the presence of chromosome, because chromosome is formed from DNA, and DNA is basically genetic material. What this genetic material is doing? Genetic material is basically for the transfer from one generation, basically parents to the offspring. So it is responsible for cellular reproduction. Okay, then the next point, it determines the cell development, maturity by directing the chemical activities of the cell. So basically it directs the chemical activities of the cell that is responsible for the development and maturity. Okay. So a nucleus is responsible ki I want to do this, I want to do that. So nucleus will be the organelle which will be directing other organelles ki hi, this is your work, you have to do this. This is your work, you have to do this. So basically it is giving the directions to the other organelles also that you have to work in such and such directions. Okay. So now we are done for the nucleus, and okay, next we will study in the later classes. Today what we have studied is for the cell wall, cell membrane, nucleus, and what are the things or what are the structure which has included in the cell, that is cell wall, plasma membrane. Cell wall not in every organelle, not in every cell. Cell wall is already included, cell wall is included in plant cell, animal cell, plant cell, bacteria cell, fungi cell. Animal cell does not have cell wall. But that is the basis of classification also. On the basis of cell wall we have two kinds of cells, animal and plant. Animal do not have cell wall, plant have cell wall. Then on the basis of nucleus we have two types, that is prokaryotic, primitive nucleus, eukaryotic, true nucleus, and true cells, everything. Okay, new type of cells. Then we have studied about the cell wall, that it is rigid, it is dead, it is not found in every cell, and plant cell consists of cellulosic cell wall, fungi consists of chitinous cell wall, and bacteria, that you need not to know, but you should be knowing that bacteria cell consists of cell wall. Then talking about plasma membrane is basically selectively living permeable biomembrane. It is formed from bilayer lipid, and proteins are inserted, or they are inserted in those lipid bilayer thing. Then this is moving, and they are responsible for the transportation mechanism from outside to the inside the cell. Okay, then lipid is formed from two things, that is head and the tail. The head part, that is water loving, which is the outside the cell or inside the surface, and in between the tails are interlocked between each other so that they do not face any water environment. Okay. Then we have studied this structure what we have seen, the ultrastructure of plasma membrane, that was fluid mosaic model. It was given by Singer and Nicholson. This was there for today, and what we have studied at last was nucleus, the brain of this. Obviously we need brain to function properly. So what we have studied is the nucleus is the brain of cell, responsible for all the cellular activities, reproduction and maturity, everything, for the guidance, because we need a higher authority to guide us that we have to do this function, we have to do that function in this direction or in that direction. So that directing thing is the nucleus, and nucleus is also for the reproduction because it consists of chromosome, which consists of genetic material transferring from one generation to another generation. Basically it passes the information from one generation to another generation, that is nucleus. For later classes we have more cell organelles, cytoplasm, and other organelles for this chapter. I hope you will go to the first lecture if you have not seen it, so that you will get to know what we have studied in the first class, then you will study in this class, and then you will come in the later classes to finish the chapter with me, so that we will be done with this chapter for the term 1. Okay, so here I am signing off. Don't forget to study after this. Okay, either take it as like proper mentor guiding, that you have to study the NCERT, each and every deep line of NCERT, because the content you should be knowing it very thoroughly, very effectively, ki anything asked, ki this is written at that page. Okay, you are aware about what is written in your books, so that you have to do, that you have to master, like you have to read your books thoroughly, only then you will be able to answer in your exam very effectively and in a very appropriate way. Okay, so here I am taking off from all you guys. I hope you enjoyed the session, and don't forget to study with buddy. Bye bye students, have a very good and amazing day ahead. Bye bye.